Happy sunny Thursday afternoon to you. Before I start, any announcements from anybody?
Donuts tomorrow at 10:30.
Donuts tomorrow, 10:30.
Where? What's that called?
Third floor kitchen.
Third floor kitchen.
Any others? All right, so this is a real treat for all of us. Earlier this week, Marjorie said how much fun it was to see the second year student talks, because we get to hear about all of the work people are going to do. Now we're hearing about all of the work people have done, but we still expect more from you. I should also say that I have an introduction I'm going to read. George Kling helped me write it. He did the first draft. You'll probably be able to tell where I have added my own bits and pieces. So it's my privilege to introduce our speaker today, Jake. You all know Jake. He received his PhD from the University of Georgia in 2013, the year his beloved Louisville Cardinals beat the University of Michigan in the NCAA basketball championship. He was then awarded a NSF postdoc fellowship that he held at the University of Washington. He did another postdoc at UC Santa Barbara, the Gauchos, and joined EEB in 2017, the year Michigan beat his beloved Louisville Cardinals in the NCAA tournament.
Michigan.
Based on his substantial contributions to science, teaching, and outreach, he was granted tenure and promoted to associate professor. Well, it's not Jake calling.
Ironically, it's my dad, though.
Right. Started here in 2017, and he was elected an early career fellow in 2020. Since then, you've all got to see him in the classroom, you've seen him in the field, you've seen him give talks, you hear the kinds of questions he asks. You know what an amazing community member he is. You should also know the Louisville Cardinals went 4-28 last year. Jake, take it away.
All right, thanks so much. So it has been, well, first of all, it's an honor and pleasure to be here because it means that I got tenure. I could be somewhere else, so. It's been good to think about this talk because I could reflect back on sort of where I've been, but also think about actually where I started when I came here, right? So it was about 7 years ago that I gave a job talk here. And so I looked back at some of those slides and I decided to start out with some of those slides. And so I started that job talk talking about patterns and process and how, talking sort of big picture, patterns and process and ecology, and how ecologists think about the way that processes form these patterns across ecosystems. And for example, these Namibian fairy circles in West Africa, you can see they're sort of non-randomly distributed across the landscape, and we see similar patterns in other ecosystems. So this is a desert landscape in southwest United States where you see the sand distributed with the vegetation in sort of these seemingly patterned manners. And likewise, we see similar patterns in ecosystems that I work more closely with. So these are patch reef ecosystems on the Great Barrier Reef. And if we zoom into one of these nodes of interactions, we see heightened interactions between features of the landscape, including the habitat characteristics, consumer behavior, and ecosystem function. So we're seeing these feedback dynamics at these individual nodes, but as we scale out, we see that these nodes are also connected spatially across the landscape by both abiotic and biotic factors, including things like fish that are moving between these landscapes. And so after having looked back at these slides and reflected on them a bit, I realized one really important thing. I had absolutely no idea what I was talking about. I was using a bunch of big words and I was cramming them together. And the worst part about this statement is that you all hired me. One person called me out at the end of the talk, and it was George. And it was when I was here. And he said, "Did you know what we were talking about?" And I basically said no, because I had to be honest. But also, while I was thinking back about time, I thought, well, where was I in my life when I was thinking about these sort of nonsensical ideas? And I found this photo of myself. That's my chicken, Lisa Ruiz. And so you hired that, basically, okay? But in thinking back in this, in the where I was at that time, I've been able to reflect on where I am now. And so really, I am actually interested in patterns and processes, but I'm really interested in the processes that lead to the patterns in ecology. And specifically, I am interested in these feedback dynamics, but more specifically, I'm interested in how they create biomass. The creation of biomass is a fundamental question in ecology, but it's also the basis of all the ecosystem services that we all rely on. And specifically, I'm interested in the drivers, which these things are, of that biomass production. And tropical coastal ecosystems are a particularly interesting system to study these questions because they're paradoxically some of the most productive ecosystem types on the planet, but they exist in extremely nutrient-poor environments. And you can see from the clear water, there's not a lot of ambient nutrients. If there's a lot of ambient nutrients, the water would be green, but there's not. But yet, despite that, they're extremely productive. So refining my questions as I get older, I've simplified them, in fact, to two basic questions. What are the drivers of productivity in tropical coastal ecosystems? And so this question allows me to ask these really fundamental questions in ecology, but then I can use those questions to direct them towards try to understand or try to improve conservation and restoration efforts. And so my second question is how can we leverage this knowledge to improve conservation efforts and sustainable food systems? So the idea is that the fish are essentially creating, or that consumers are creating these feedback dynamics within these ecosystems. And by understanding the drivers of those productivity, we can actually take that knowledge and interject it into restoration and conservation to facilitate those feedbacks in a more sustainable way. But importantly, also reflecting back sort of on my sort of journey and how I go about science, I've come to realize that my approach to doing science might be as important as the science I actually do. So I work a lot in these coastal communities, and by going to the field and working directly with these local communities, I'm engaging in with them directly and into their livelihoods to understand these systems a lot better. So a lot of this was essentially born was born out of sort of my simple interest in communities. So I go into these local communities because I'm just interested in understanding about who's there, who's living there, and why. I'm also extremely interested in drinking beer. So it's a really easy thing to do. You go into local communities, the first thing you do, you sit down on the stoop and you hang out and you just get to know the folks in the area. But there's also a really important need in doing this, right? So this work directly relies on, the work that I do in these local regions relies directly on the support of these communities. So it's critical to gather local knowledge to first understand things like where are the fish? How are they fishing? How has fishing been over the years? Where is the seagrass, right? So I rely directly on the resources of the local knowledge of these communities. And I need the physical help, right? So we're doing these pretty high-level experiments where we're hauling a bunch of blocks around, and so we actually need physical help. And so what we do is we pay local fishers to use their resources and their human power to actually do the research in and of itself. And this can also provide a little bit of income in a lot of these lower income communities. And what this does, this engagement, creates community buy-in. So basically, by engaging with the local people, showing that you respect them and their understanding of the system, providing a little bit of resources to them, we get buy-in in the science that we do, which ultimately can lead into greater support into conservation in these regions that actually really need it. So what this does is it helps me actually hone my research to tackle issues that matter to local communities, right? Again, I said that we're testing basic fundamental science, but we're trying to direct it towards actually the conservation that matters in the place that we're working. And this engagement really allows us to do that. So essentially how we engage communities can actually create positive feedbacks in and of themselves that both benefit the science, but also hopefully benefit the local communities that we work with. So, excuse me. The home crowd is like a different situation. You roll up in a new place, it's like, God, it's like, okay. So currently there's 2.5 billion people that live within 100 kilometers of a coastline. Most of these people live in tropical regions and in impoverished conditions. And by 2050, these numbers are expected to increase by almost twofold. So a really big challenge that we face is understanding the mechanisms that promote sustainable food security for these coastal communities. And I stress food security because it's my opinion that people that aren't hungry can afford to make better decisions. And in fact, the ecosystems that we work in, coral reefs, seagrass beds, and mangroves, are essential for food security, but they're also among the most impaired ecosystem types globally. This is actually an image of two systems that I've worked in. This is in the Bahamas, where the reef is actually doing quite well and very healthy. This is an image in Haiti, where all these nubs are old Acropora palmata, which is the primary reef-building coral in these ecosystems. And importantly, these are among the most productive ecosystem types on the planet. And in particular, they have extremely high rates of primary production. In particular, these two, seagrass beds and mangroves, have the most highest rates of primary reduction of any ecosystem type on the planet, which allows them to sequester more carbon per unit area than any ecosystem type in the planet. And this high rates of primary production fuels high rates of secondary production, which in turn provides food resources for communities that live within these ecosystems. And so our goal is to understand how these factors interact, but to include communities and humans within the broader picture of the ecosystem. So in the late 1800s, the agricultural revolution was responsible for pulling billions of people out of starvation. And the agricultural revolution was born from science, very basic science, where we understood, or we developed the understanding that nutrients could be used as fertilizer to increase production rates and actually create more food resources. And this literally saved billions of people from starvation. But there were repercussions to this process. And we see by spreading all the fertilizer across these ecosystems, it's having drastic impacts in terrestrial ecosystems, but it's also having really negative repercussions in aquatic systems. So this is actually, in my opinion, a beautiful image. It's an algal bloom at the delta of the Mississippi. These algal blooms are due to the high rates of fertilization or fertilizer that we're dumping on our crops, and they cause immense dead zones in these coastal areas. So they're killing off fisheries and degrading these ecosystems in general. In our system, we may have an opportunity to take advantage of a different source of fertilizer, and that's from fish. So in the early 80s, Judy Meyer showed that fish grunts aggregating around a coral head could actually increase the production of that coral by up to 70%. And the idea here is that the grunts are providing nutrients that's essentially fertilizing the production of the coral. And in this sense, it's essentially they're creating or they're farming their own habitat because the coral is essential for them for shelter. So, and basically this study alone inspired basically all of my PhD work, during which I was able to essentially find that fish are among the largest sources of nutrients to coral reefs. And so building on that idea and building on the fact that these fish are inherently facilitating their own habitat, my question is, can we actually initiate these same feedback dynamics to restore coastal tropical ecosystems? So I essentially am talking about farming the oceans. So the idea is relatively simple. We can build structure. And so here we always use cinder block, which is made of limestone, which comes from coral in the same system. So it's a very fish and coral friendly habitat, right? So we build this structure, and fish aggregate around that structure. They recycle nutrients locally to the ecosystem. And important to what we're doing, we actually build it always in seagrass beds. And what this increased fertilization from the fish excretion does is it enhances primary production, which in turn can provide more food and resources or food and habitat for invertebrates, which in turn can feed back and potentially augment the fish populations as well. And so this idea we've been working with for now, I think it's like 14 years. And this is the first set of reefs we built. You can see they're kind of dinky. They're not glued together, but there's lots of fish, right? There's lots of fish, high diversity. And with this study, we found something very simple. And that is, if you have more fish, you have more nutrients, you have more primary production, right? So each data point on these plots represents a different community on a different artificial reef. Communities that have more biomass have more phosphorus supply and nitrogen supply, and that in turn creates greater seagrass growth. And we've since studied this pattern over and over throughout many different ecosystem types in many different ways and found it to be very, very consistent, right? So we've studied it across different areas and on different conditions, et cetera. So what I'm now interested in, or I guess what I'll be talking about today specifically, is so that pattern that we've seen emerge where we get heightened production around the reefs. what are the specific mechanisms that actually underpin the increase in primary production around these artificial reefs? And so this is, again, is the sort of fundamental question that underpins the work that we're doing. But then in turn, what are the ecosystem level implications of this increased local production? So can we actually scale these up to be at scales that influence communities in a positive way, right? That's sort of a sneak peek to what I'll be talking about at the end. This is a study that there's lots of fish on, obviously. All right, so I talked about how we can enhance agriculture by adding fertilizer, right? And this is the science. The science has underpinned this knowledge by showing us this can happen. Well, there's multiple mechanisms by which this can occur. We can have a situation where any increase in a single nutrient will enhance primary production. So the green here will be primary production. So the more phosphorus you have under any condition of nitrogen, you're going to get more primary production. Or the more nitrogen you have under any condition of phosphorus, you can get more primary production. So it's the supply rate in this case would be enhancing production. Alternatively, it could be a ratio thing. You need a certain amount of phosphorus and nitrogen at certain ratios to be able to actually affect that primary production. So it's not necessarily about the supply rate of one of them, but it's the ratio. And in fact, this is widely supported in a lot of the literature in theory that this should be a mechanism that should be underpinning primary production. It's sort of the basis of ecological stoichiometry. And it's something that I sort of fell into and really became obsessed with these ideas during my PhD. And this was supported by a really important finding that I found towards the end of my PhD, and that was specifically that fish communities on coral reefs were actually supplying nutrients at the rate that was optimal for coral growth. And so basically what we found is this is just an axis of nitrogen to phosphorus ratio. So down here we have more phosphorus. Up here we have more nitrogen. And what we did was we used surveys of coral reef fish communities across the northern Caribbean. These are generally healthy fish communities. And we estimated how much that community was supplying nutrients to the system, but also what their ratio was. And across 122 different communities that had vastly different biomass, vastly different species composition, and vastly different amounts of nutrients they were supplying to the ecosystem, they supplied a really consistent ratio of those nutrients to coral reefs. And this was really peculiar to me, and so I started looking into the research, and I thought to see under what environmental conditions coral reef were found to grow best. And the literature showed that when the coral were enriched with ratios that were either really high in N, so this actually mimics or looks like fertilizer input from ag fields, or really low or really high in phosphorus, which is more like sewage, the coral always died. But the only situations in which the coral did well was when it matched perfectly with the ratio that we're finding that these fish were supplying at these really large spatial ecosystem scales. Right? So it's a super surprising finding, super fascinating, and provides a lot of support that the ratio should make a big difference in this context. So we know roughly what that the ratio of nutrient supply from fishes coming in these ecosystems, that could be the key thing that's facilitating the habitat in which they persist. But we also know that humans are adding nutrients to these ecosystems, typically at a very different ratio than what we were finding with the fish excretion. We also have shown that with increased human population, we see drastic reductions in the amount of nutrients that are supplied by these fish communities. In fact, comparing ecosystems that are fished versus coral reef ecosystems that are fished against that aren't, it's about a 50% reduction in nutrients supplied by the fish community. Again, this is one of the largest sources of nutrients to these ecosystems in a really nutrient-poor system. So that's probably a very substantial effect on these systems. But we also know that where we have more humans, we have more human-mediated nutrients entering this ecosystem. So an important question is, how do changes in supply rates and ratios from different nutrient sources affect primary production? So to test this, we constructed a series of artificial reefs that we manipulated to simulate fishing pressure. We did that by literally filling in the blocks to minimize the amount of habitat, which reduced the biomass by a lot. And we also enriched the ecosystem using these little fertilizer tubes that had nutrients that were had very high amounts of phosphorus relative to nitrogen, so they simulated fertilizer. And what this did was, and so it was a fully factorial experiment across 16 different reefs, and what this did was it provided us a really nice gradient of both nutrient supply rate, but also the ratio, right? So we're simulating fertilizer and fish in these systems. And then we could test those effects on the seagrass itself, but also on coral. And so I'm going to show you all pretty quickly just the results of these findings, and then kind of keep moving forward. So with respect to the seagrass, what we found was that so here we have the growth rate of the seagrass on the y-axis, distance away from the reef. So we measure things as a function of distance away from the reef, and you can see that as things get further from that source of nutrients, you get less primary production. But importantly, the treatments in which we had high nutrients from the fertilizer and either high fish or low fish, we get these really strong effects on seagrass production, right? And so basically what we're finding is wherever we had the most nutrients, seagrass grew more, which is sort of obvious in some way. But we actually found no evidence whatsoever that the ratio of nutrients affected primary production at all. It was all about the supply rate, right? So this doesn't conform to some of the theoretical expectations or what sort of we were thinking we were finding previously. With respect to coral, we simulated, or we used the same reefs and added coral nubbins using 2 different species of coral hanging on a coral tree across all the different reefs. This work was done in collaboration with an old graduate student buddy of mine, Dusty Kemp. And I'm not going to get into the nitty-gritty details about this, but in very short, we found that, to our surprise, so the expectation is that nutrient enrichment should actually kill coral, right? But instead, we found in all conditions, the coral grew like hell, right? We had extremely low mortality, which is not really even common for a transplantation study in the first place. Typically, in the best situations, you have low mortality. And we actually found no effect at all of the nutrient ratios, right? So that's strike 2 for nutrient ratios in this context, suggesting that theoretical that theory is not really holding true. And so I'd like to note that Nepsis Garcia is a new PhD student in our lab, and she's going to be sort of carrying this coral torch forward. We're not exactly sure how, but pretty excited about that work we're going to get together to do. All right, so that's all fine and good on sort of an experimental scale, but we're really interested in how these things actually work in the real world. And so in, I think it was 2014, Craig Lehman and I established a different study where we actually went to the places that were being affected by these perturbations. So we had we built a series of artificial reefs on the island of Île La Vache in Haiti, where we have extremely high fishing pressure. And when I say extremely high fishing pressure, I've never seen anything like it in terms of the number of traps and the intensity that fishing was happening in this region. So I would say some of the highest in the world. But there was also extremely high nutrient input into these ecosystems. So it's very common for one of these communities, which could have thousands of people in it, to not have a single toilet. So the only place to go to the bathroom is either the mangroves or the ocean, right? So extremely high rates of fishing pressure and nutrient input. And then we built a handful of reefs in the Bahamas where we had a sort of gradient of both. A lot less fishing pressure, but we had some nutrient input in these systems. So these are 2 reefs that we built across the gradient, and to our tremendous surprise, this reef at the bottom, where there's the most fish, is actually the reef in Haiti. There's actually one of the reefs in Haiti, right? So we're seeing this really high biomass of fish on the reefs in Haiti. And the Bahamas, that's still quite a few fish. They're a lot larger in size. And you can see from these frequency histograms, in the Bahamas, we got a lot larger fish, which is expected, right? If there's not a lot of fishing pressure, you just see larger fish because fishers select for the larger fish in general. So each line represents a different artificial reef, and then the shaded in line represents the average of the total. But in Haiti, we see a lot of fish, high densities, but they're really truncated to a very small size, right? And what we think is happening, we couldn't test this, but what we think is happening is those sizes are the ones that are actually able to escape through the holes and the traps and the nets, right? And so it's sort of a safe haven for these fish that are able to get away from the fishing. Plus, just because of the gear that the local fishers use, they have a hard time actually fishing on these reefs. So they're not very effective at fishing the reefs themselves. And so what this translates to is we get extremely high biomass. So green is Haiti, and these are all the different reefs we use. So the high biomass of fish on the reefs in Haiti, much higher than those in the Bahamas. And in turn, we get extremely high rates of nutrient recycling in terms of phosphorus and nitrogen relative to that of the Bahamas. So this is totally counterintuitive, right? We would never have expected to see this because we would have assumed there'd be no fish in Haiti, right? But instead we're seeing tremendously high rates of nutrients coming into these systems. And, but we did see the effect of the nutrient input, so sewage input in the system was likewise extremely high, and you can detect that in the sediment, both phosphorus and nitrogen. So again, the green dots are the reefs in Haiti, the purple are those in the Bahamas. And in particular, you see some of the sites that are closest to the communities had really high sediment nitrogen, but all the reefs regions had really high phosphorus. And again, that's that phosphorus signal from the sewage, right? So we're getting really high inputs in Haiti from fish, really high inputs in Haiti from nutrients. And in that case, the expectation could be that you actually would see negative effects on the seagrass. But in fact, we saw the opposite, and we saw the seagrass just growing like crazy, right? So this is a plot showing, again, all the reefs in the different points. And we're looking at seagrass growth rate and then the carbon production of the seagrass per unit area per time. And you can see in Haiti, the seagrass is just growing like crazy, at times, 3 times more than those in the Bahamas. And importantly, the production was actually so high that it became carbon limited. I just think that's a wild stat. I'd never actually heard of seagrass or any plant becoming carbon limited in that sense, but apparently it does happen, and it did happen there. But there was actually no evidence, again, that the ratio of nutrient supply affected seagrass production at all, right? So wherever, even though we're seeing these, even though they're having these massive impacts on these ecosystems, essentially the seagrass was resistant to these stressors in Haiti and still growing like wild. So I think that these sort of unexpected findings tell us a few really important things. In particular, that we actually need to go to the sites that are being impacted and study them in situ, right? So we wouldn't have expected to see reefs look like this just thinking that Haiti is highly overfished. If we were just to have modeled this without actually going, we would never have found what we found, right? And so I think it's really important to actually go physically to these sites for these reasons to understand what's going on, because they're extremely complicated systems. But then again, in doing so, we're engaging with these local communities where the conservation actually has to happen, right? And so by engaging in these communities, we're engaging them in the science directly, and that's facilitating and opening doors for potential conservation action to occur. So basically what we found in those series of experiments is that what's really, the mechanisms that are really driving the production of these, the primary production of these systems is really just the amount of nutrients, which contrast theory and really was kind of has continually blown me away. I've always been surprised that we're never seeing that signal. We are seeing, when there are high rates of nutrients, we do sometimes see a slight shift in the community composition, which is important, but it's still highly dominated by that one species, Thalassia testidium. So the next steps are really to understand the mechanisms then of how this primary production translates into production in the invertebrate biomass, but also into the fish community. And that's exactly what we're doing. So Bridget Shaika is a PhD student who's finishing up in our lab next term. She's doing an amazing job taking everything we've done a step further and asking these questions in the below ground biomass where all the real carbon sequestration actually occurs. Shreyan Seth is a sort of all-star undergrad student who's taken on the mantle of understanding how the primary production is actually feeding into the production in the invertebrate community. And then Katrina Munsterman, who's a PhD student, is trying to understand how that energy then gets into fish production. But I want to take the opportunity to sort of give a shout out to this sort of army of undergraduates we have working for us. And another thing that I've learned sort of reflecting about my time here is how amazing it is to work with undergrads and how lucky we are to have such amazing undergrads. So these undergrads, all the 5 students here are all leading or have published first author papers in my lab, right? So it's just a pleasure to work with these sort of amazing students, and I want to give them a shout out for sure. So this is sort of an anecdotal thing that's actually quite interesting. So the idea of artificial reefs is very not new. It's been going on. People have been using them for thousands of years, literally. And one idea that people have been using lately is we have all these oil rigs in the ocean, and they provide habitat. And as we dismantle them, because they extract all the oil, you have to remove them. People are saying, well, don't remove them. Take the top off and leave them. And this idea is rigs to reefs is what they call it. And a study that came out in 2014 showed that, that these rigs were actually the most productive. They had the highest fish production in the ocean, right? So it's a really fantastic study. I've put a lot of energy in trying to understand it. It's a really great study, and it's pretty wild finding, and I was super surprised by that. We are also interested in understanding the productivity of the fish on our reefs. And so using similar models, which I think we've actually improved, Katrina Munsterman has taken, conducted similar estimates on our reefs across the Bahamas and Haiti, and this is the production we're seeing on our reefs. So it's substantially higher. And this, I want to point out, so this is the base, the midwater, and the complete. So this actually is an aerial quantity of about 45 feet of structure. Our reefs are in 10 feet of water, right? So we're seeing, whatever, how many more times that is, right? So we're seeing extremely high rates of production on these reefs, which is really cool. But it does beg the question, do artificial reefs simply attract fish, or do they actually create new production, right? Are they just drawing fish from somewhere else, or is there actually new production happening? And so this leads me to the sort of second part of the talk, is what are the ecosystem level implications of increased local production? So we can think about this from, sort of outline it through a series of simple hypotheses. So in this scenario, we have, say, a hectare of seagrass. You have some fish swimming around. They're doing their thing. You get a certain level of primary production within that system. If we plop a reef down in the middle of that, the fish will aggregate to that reef. And we've shown time and again that what's going to happen is you get increased primary production around that reef. But because they're no longer swimming in that open seagrass, maybe there's a detriment to the seagrass, and it's going to reduce in primary production. And so you essentially get no net change. An alternative hypothesis is that because as they're swimming around in the open, they're distributing it really sort of all over the place, and that's not having a really substantial effect on that open seagrass. But we know you get that big bump in production around the reef, and so you could possibly imagine a scenario in which you actually get a heightened amount of production within this system. So I've been thinking about this for a long time, and it's actually a really important question, because if artificial reefs are really just attracting fish, that's actually a bad thing, because then people could just overfish it, right? So we really need to know this to actually begin to employ these things in a conservation setting. And we to this date we do now, because what I'm about to show you, don't have a good answer. But it's extremely difficult to test empirically, which is really frustrating to me. So what we've done is we've turned to models. And I've already been given shit by John about not explaining the details of my models. I'm not going to explain the details of this model. I'm not going to get into the equations. But what we used was an individual-based model to actually try to explore the ecosystem level implications of consumer behavior by aggregating around these reefs. And so this is work that was conducted with Kenzo Esquivel, who is a master's frontier student, who's the first student in our lab, and Max Hesselbart, who's a sort of postdoc extraordinaire, amazing modeler, and we generated this package called R. I think that was Max. I'm not sure you can remember that one. So the idea is relatively simple. We have a seagrass ecosystem. The primary production is underpinned by a single nutrient production model. And what's really awesome about this model is that it's underpinned by a ton of data from our system that we collected, right? So we can really trust this model, and in fact, it really does mirror what we see in the field, right? So these models are matching the rates that we're seeing of production of seagrass in the system. And importantly, it allows us to quantify above ground and below ground processes that actually talk to each other in the model environment. And swimming around, the individuals in the model are the fish, and they're underpinned by a bioenergetics model, which also are parameterized by a bunch of empirical data that we have generated in our lab over time, both in terms of the movement, but also in terms of the physiology of the animals. And so what this allows us to do is allows us to quantify ecosystem scale above ground, below ground, and total production of the primary producers within these ecosystems. And so what we're interested in doing is just testing the effect. For this paper in particular was very simple. We just wanted to test the effect of behavior, of aggregating behavior on the reef for ecosystem scale nutrient production. And so the way the IBMs work, it's sort of like Pac-Man. It's like a video game. It's a simulation model. Here, the fish are just swimming around totally randomly. Here, they're actually attracted to the reef. It doesn't look so much like it because they're swimming so fast, but you can really see it clearly in the footprint of nutrients. So this is where the nutrients are distributed over time when they're swimming around randomly. And then when they're aggregated to their behavior is aggregating around the reef, you can see that it's highly concentrated in the center of the system. So what did we find? This is a little bit of a complicated figure, but all of my figures seem to be complicated. So in blue, if the data points are in blue, it shows increased production with fish attraction. If it's in red, it shows reduced production with fish attraction. Starting capacity biomass just means the level of enrichment we started the system off at. And then we ran the model with different numbers of fish, different numbers of fish in them. And this is actually a simplified version of the total study, but this will work for now. So what we found was that with respect to above ground production, we always saw increased above ground production when the fish were attracted to the reef, right? So basically we see that bump around the reef every time, which is sort of expected and what we've been seeing in the field time and again. With respect to below ground production, we actually did see some loss in production in the open seagrass because the fish weren't foraging out there as much and they weren't providing those nutrients. But importantly, if you look at the scales here, it's actually relatively small. So going from 0 to 0.05 versus 0 to 0.5, right? And so the net effect is that we see an increase in total production at the ecosystem scale just from the fish aggregating around the reef. So again, just to be fully clear, these are closed systems. The only difference, they have the same amount of nutrients in each system. The only difference is the behavior of the fish, that they just tend to aggregate around the reef. And with that, even though there's the same amount of nutrients, we still get substantially greater production in the system due to that aggregating behavior. And the reason that's occurring is because there's a synergistic effect associated with uptake dynamics. So basically, bigger seagrass can take up more nutrients and become more productive, right? So we're sort of getting more out of the same, essentially. And so in my opinion, this is super exciting for conservation. First, it's actually the first case where anyone's shown that there is novel production happening on artificial reefs. But importantly, that production can be used as a sort of mechanism to enhance carbon sequestration in seagrass. Again, seagrass beds have the highest rates of carbon sequestration of any ecosystem type on the planet. And then those nutrients can, in turn, potentially fuel fish production within these systems. And so with this kind of information, where my head goes is something like this, right? We can actually think about implementing these things in space to actually use them for promoting these ecosystem services that we care about. But importantly, if we're going to do something like that, we still have to manage these systems. And so a broader question is, can we manage these artificial reefs across space to actually increase the resilience and stabilize these services over time? So importantly, these ecosystems are being constantly affected by perturbations, in particular, storm events. So this is an image of a picture I took through my windshield of Hurricane Dorian, the impacts of Hurricane Dorian. These all used to be buildings. This is in Marsh Harbor where I work. So the impact, the frequency and the magnitude of storm events in tropical systems is increasing. And people think about the magnitude with this is one of the largest storms in the history of the Atlantic, but actually the frequency is the one that's going up more. So we're just seeing more storm events. And with each of these storm events, you get disproportionate effects of nutrients coming into the system because they're just flushing out of the terrestrial ecosystem into the ocean, right? And so if we think about one of these systems and think about the production over time, we may see these highly erratic rates of production due to different amounts of nutrients coming into that system over time. And if we scale up that thinking and start thinking about how these reefs relate to one another, we see a series of the reefs could be affected by storms differentially affecting production. And if that production is asynchronous, right, so if it's not happening all the same way, then what happens is we actually stabilize production on the larger system. And so this is simply the portfolio effect. And the portfolio effect is a concept that was born in economics, and the idea is really simple. You have a bunch of money, you want to invest it. You don't invest it all in Amazon, because if Amazon takes a big shock, all your money goes down. So you invest in a diverse portfolio of different businesses, and that means that they can all take the shocks differentially, and that can stabilize your returns over time. So the same concept applies spatially, where spatial diversification can maximize the stability of the whole. And in doing so, it can increase resilience against these perturbations. So Max and I got back to this model and wanted to specifically test this. And so we were asked essentially the question, can we manage for resilience in the face of unpredictable perturbations that we're going to be seeing increasingly occurring within these systems. And so the idea was simply that we took the initial model, and instead of being closed, we opened it up, and we linked it spatially across, well, space. And we had different amounts of nutrient enrichment coming in from the system, both spatially and temporally. We varied those in terms of the amount that came into the system. And then we had fish nutrients being supplied throughout the system. So fish were also moving throughout all these different systems. And we were varying those fish in terms of the population size. So we could essentially simulate the effects of nutrient enrichment coming in at different times, and then also fishing in terms of the fish abundances. So we were first interested in looking at just how these things acted independently. So in terms of fish dynamics alone and nutrient enrichment alone. And the y-axis here is the portfolio effect, so you can sort of think about it as a proxy for resilience. And you can see that fish dynamics have a substantially greater effect on the portfolio effect or resilience of the system. Right? So it's all about the way that they're moving across the system, but also importantly the biomass of those fish. But importantly and more realistically, thinking about them together, because they don't ever happen in isolation, we found this really interesting synergistic effect where high population sizes of fish and low amounts of nutrient enrichment created a synergistic effect and stabilized these systems substantially more than the other combination, right? And so this is actually sort of intuitive in the sense that the traditional management says we should protect fish populations and we should reduce nutrient enrichment. But it's actually really cool to see that if we do this simultaneously, we actually get this synergistic effect that is actually increasing the resilience in these systems. So what this is telling us is essentially that fish are a clear management priority, right? If we're going to pick one, we're probably going to pick fish. But we shouldn't be doing that in isolation. We still need to think about nutrient enrichment into these ecosystems. And it also is really saying that this artificial reef may be a useful tool in sort of small-scale management initiatives, right? If we can actually put them together and couple them, they may actually help stabilize some of the services that we actually care about. Again, carbon sequestration, fisheries production within these ecosystems. All right, so what have we learned and where are we going? So first, we've learned that fish initiate feedback dynamics that fuel high rates of production in these tropical coastal ecosystems. So that's the process that we've been trying to understand the basis of. The patterns that we're seeing is this process is really predictable around these artificial reefs, and it can increase local and ecosystem scale production. What we need to do now is really understand how this actually translates into the fish production and how we can scale these processes up to be relevant for conservation. And this remains really the key challenge. So the way we're doing this, or one approach in which we're doing this, is we're trying to figure out is there sort of an optimal design that can actually enhance fish production. And the idea is relatively simple. We can build more reefs, essentially. So this is now clusters of reefs. We can space them out just by a small amount, so that's 1 meter spacing, which simulates more like a coral reef ecosystem, which in turn actually provides more shelter for the fish that prevents them from being fed on by predators, which includes humans. These would be very difficult to fish by humans. Or we can sort of maximize the fish pee angle by spreading out the reefs. And what that does is it actually creates a synergistic effect that we've measured in the middle of these reefs, whereby the fish pee from this reef and this reef actually jacks up that primary production in the middle just as much as it does right next to the reefs, right? So you get this synergistic sort of bottom-up effect. And what this allows us to do is to test the mechanisms that are actually driving fish production within these ecosystems, but doing so at scales that are actually relevant for conservation. So one single reef is not going to feed a community. This is actually getting a little bit closer to what actually might be useful for humans. And we're doing this in collaboration with the local fishing community in the Bahamas, where we actually built all the reefs with them, and they're actively fishing these reefs. And so we're actually seeing how fishing pressure may change these dynamics that we're seeing in a different system, right? So this has been replicated. So we have in the north of the Bahamas and the south of the Bahamas. So we started this experiment, I don't even know when, 21 maybe? 22? It's so hard, we have bad permitting issues, so I lose track. So this is an image of the reefs that are separated by 1 meter. They kind of look like marching soldiers or something. This is right after we built them. These are the reefs that are separated by 3 meters, and these are reefs that are separated by 5 meters. The footprint of the 5-meter reef is so big you have a hard time sort of getting it on camera, essentially. And interestingly enough, the reefs that are being fished actually have a lot of fish on them. I don't think it's the same mechanism for the reason why we see the reefs in Haiti having a lot of fish. These videos show a lot of lobster. Those lobster are all gone because they are actually fished out. In contrast, the areas that are not fished do have quite a few bigger fish, but it's quite different. I think something else is at play, and I think this also illustrates why working in the system and doing the work there is really important because things are crazy. So so the way that I'm thinking is that we kind of need to move away from this model, right? So this model basically says, this is Île aux Vaches island I worked in Haiti. These people depend on fish. They live on the resources in their oceans. You can't go into this place and say, hey, y'all can't fish here anymore. This is now a marine protected area. That's not going to work in any context. They're not going to do it, even if you told them to do it, right? So we have to get away from this model. And sort of what I'm proposing is move towards something like this model. So instead of saying you can't fish the whole thing, work with local communities, which before I left, I was working, and we were actually going to implement one of these in Haiti, but I haven't been able to go back because of the unrest. But basically, we could protect an area maybe the size of this room, which is not going to hurt anyone's feelings, right? That's not very much to not fish, and build these throughout the system, and maybe use sort of adaptive management. We can rotate which is fish, which is not, et cetera, et cetera. There's lots of ways we can think about this. And so I think that this is a really interesting way to think about moving forward. And importantly, it's really critical that while doing it, we're engaging with these local communities, right? We're adjusting how we think about these ecosystems based on what we learn from our work with these local communities. And one of the ways that I've been doing this, along with my lab, who gets a lot of credit, oh, that's a picture of moose. That's funny. That's my dog. He's not supposed to be there. It was like kids here having a good time. Instead, you see my dog. That's funny. Okay. So I've developed, in collaboration with my lab, we created this outreach program called PODS. It's a pre-college ocean discovery and science program. And basically what I've learned is by working in these different communities throughout the Caribbean, you often don't see the faces of the people that live there be the faces of the people that are leading the conservation initiatives locally, right? And that's something that needs to change. We need to have more local leaders in these positions. And so the goal of this program is to try to facilitate that by working with local high school students. We have a sort of a short course that we do virtually, then we have a field course in person, and then we actually help them apply to college. And we actually had our first student was accepted to college like 2 weeks ago from this program. So this is an ongoing program we've been doing for 3 years now. And it's just it's a pleasure to be part of. And sort of as I guess sort of with that, I'd like to thank, that's the picture of Moose. I'd like to thank my family. They really supported me through all this. I certainly wouldn't be here without them. And that's I don't have the words to say for that, I guess. And then I would like to also thank Mr. Moose and Lucinda, who I spend probably more time with than anybody else. And with that, oh, sorry. So that's local Haitian music. That's reading. So I do want to give a shout out. I've had tremendous membership here at the University of Michigan. George, John Zach, Mark Hunter, Nate, and Deborah have been really fantastic, and have really helped me get having gotten tenure a lot that they deserve. And then I have to thank Fields, Pictures of Ecology, Tobacco, Mr. Curry, a local nonprofit that I work
We'll ask you all to leave the room. Jake will be in this committee.
That's what it felt like. God.
Here's time for questions, please.
Amazing. I'm wondering if
get information, quantitative information from them about their catch, and if you then use that in understanding like the total production on these systems?
No, we don't. We have a plan to try to do that in the Bahamas, where there is like a few fish houses. They're like the sort of linchpins, or I don't know what the right word is, but they're like the bottleneck where fish have to go through. And we can actually survey those fish houses, and you probably get a good estimate, particularly in this one region, because they're fishing that region. And so that's something I would really love to do. That would require a collaboration with the social scientists, which I have that collaboration, but we just haven't gotten to do it. But in a lot of places, they just don't have those numbers. I'd say that most of the places I work, that doesn't exist. We're starting some projects in the DR, the Dominican Republic, and I will be interested to see if they do have some of those numbers, but I don't think they will either. But it's a really good question, and it's something that it would be great to have, but yeah, we don't have it. Dan?
Following up on some of the themes from the first part of your talk, so do you think that given that there's a pretty strong overall relationship between fish essentially fertilizing the reefs and therefore essentially enhancing productivity locally, do you think there is evidence for some kind of evolutionary feedback where reefs themselves or different species of corals, for example, are structurally attracting communities of fishes, for example, to enhance their own fitness?
I mean, honestly, I do think that is true. I think that the signal is so weak that you can't pick it up with ecology. But I actually do think that is true. The ratio thing that we found is just mind-blowing. Like, that is like so, I mean, the differences could be triple in the amount of nutrients, the supply rates, but yet the same ratio, that's just weird, right? Like, and I reran it's like empirical data, but like I did it a bunch of different ways, so I was like, there's no way, that's just, I'm doing something wrong with my stats, or I don't know. But so I actually do think that there is that signal. And that's what I was going for, because I was like, I thought with the coral stay we would definitely pick it up, and there was just nothing there. It was wild. Yeah, so good question. Renan?
Excuse me, a stupid question, Jake, but I was wondering about that predominance of small fishes in the highly fished areas in Haiti. I was wondering if that could also be possibly due to changes to shifts in life history, due to the fishing pressure actually shifting the entire population towards it earlier age of maturity or something.
Yeah, I don't know. It's definitely not a dumb question. It'd be cool to know how reproductive those fish are, because the grunts actually can be reproductive at a pretty small size. The thing that's weird is that like you don't see fish anywhere else. So I would show a fisher that video, and I swear every one of them would just drop their jaw, because if they were younger than 40, they've never seen an aggregation of fish that big their entire life. Like no reef would you ever see a fish aggregating like that. So it's like, I don't know. It's really confusing. I mean, we just don't know where those fish are even come from. I think there is a high supply of fish to the system.
Somewhere.
Yeah, I think that they're coming from somewhere, like maybe deep water refugia, because the fishers can't afford line, so they can't fish deep. So maybe there's something going on with that. But again, like it's a bummer. Like the last time I was there, I had a meeting with a group of fishers in a room. It was probably similar to this, a number of fishers, and they all agreed to do one of these projects with me where we would build clusters and they would not fish one. And then I haven't gone back because of the political unrest. So, and that was 5 years ago or something now. So it's a bummer because it's just an amazing system. It's super cool and awesome place to work. Yeah.
How are you choosing where to place the artificial reef? Like, is this a field of dream scenario? Like, you build the reef and you expect fish to show up, or are you picking just a seagrass bed that's known for some activity?
That's a good question. It's sort of, I think the field of dreams thing is about right. Like, we don't really know. We have one study that shows if it's like proximity to other sort of structure, you do get certain or different rates of colonization. I think the community will stabilize to be similar. In some places we build them, there's just no fish. Like there's 4 reefs in the Bahamas where there's like 4 fish on them. And I'm like, where are the fish? Like, what is going on here? It's like really close to where there's lots of fish over here. And that's the thing. That's why I really think like we have to be working these systems, because I have no idea, and I might never know that. Like, that's just a total mystery. And so it's a little willy-nilly. And like the ones in the north with all the fish on it, I would never have expected that, but I think that is because of their proximity to really productive mangroves. And so like that's triggered me on a whole new thing. Now I'm obsessed with mangroves. I'm like, oh, it's got to be the mangroves. So, I don't know. Yeah.
Nice talk, Jake. I keep wondering about how difficult it is to study nutrient cycling in a liquid medium. And you sort of answered a lot of that question, but I think, is your modeling effort moving toward trying to understand tidal effects, like, you know, how fast water moves through a system, how rapidly it's flushed, in which directions? Because that might explain part of the no fish thing. I mean, if some of these complexes of artificial reefs are getting flushed really fast, the pea's moving away. And do you measure that? Do you measure tidal dynamics?
We don't. It's not. I'd say across all the reefs, there's not a huge variation in that. That's not a huge thing. I mean, the hydrology definitely is important, but we don't see a big variation in that happening. We do see sometimes, for example, one of the reefs was a little bit closer to the channel, and so it has a little bit more channel going into this embayment that I work in, and there is actually a little bit of directionality of the growth of the seagrass. It's a little bit more. It's a little less round, and it's like in the flow of the. It's sort of more along those lines. So there's definitely something going on there, but we're not in those really high flow environments anyhow. But it is challenging. I agree.
Last question.
Oh, okay. I love really naughty science. How much? I'm curious. How much do herbivores eat of seagrass? Like what? Like 10% of the biomass?
Oh,
Seagrass biomass?
no.
Do you have a feeling? So they're not
we can
that
quantify it. Yeah, nowhere close to that.
Productivity at all.
No, they're not at all. So I get hammered on that in review, and so we've got all of the ways to counter that. So we count bite marks on grass,
Yeah.
and we can quantify it, and it's nowhere close to 10%. I would say it's, well, Bridget. I'd say it's like 1% or something, but yeah, it's not. And it's rarely significant across treatments, and there's basically there's no clear herbivory regulation. So turtles can come through and mow some stuff down. And in the Bahamas, they're protected, and so they actually are overgrazing now. But they actually don't come around our reefs that much, which is weird. I don't really know why. Maybe they haven't found them. But there's lots of turtles there.
So as you all know, it's a long-standing tradition in EEB that new associate professors buy the first round. But let's thank the Purvis Ellison of Coastal Ecosystem Ecology, Jake Allgeier.
No one got that.